Hi, my name is Rohan Shah, and I'm a field application specialist with Thermofisher Scientific. I'm going to talk about our animal health specific molecular testing workflow today. So this is just the objective slide covering the following topics: the overall principle of the animal health workflow, the first step in the workflow called sample preparation, then the targeting component of our workflow that uses real-time PCR. Then we will talk about how to make sense of real-time PCR data and some of the challenges associated with that. So pathogen detection in animal samples comes with a host of different challenges. There's always a need to detect smaller and smaller amounts of pathogenic DNA or RNA in a sample to get the best chance of early detection. This is made more difficult due to the organic complexity of some sample types, like oral fluid and feces, that could contain several inhibitors of downstream detection technologies like qPCR. Most labs test a variety of sample types and pathogens, so it's challenging to have workflows that cater to all those sample and pathogen types. Timely results are also necessary so that the lab can act based on accurate and timely data. Thermo Fisher is committed to providing dependable and flexible workflow solutions, and these include reagents as well as instrumentation and software to assist the lab in molecular testing of several animal pathogens found in a variety of sample matrices. We also have some government licensed products that can help ease regulatory challenges that might crop up. Our qPCR workflow solution is shown in the graphic here. We provide reagents and products from sample collection all the way to qPCR data analysis. For instance, our sample preparation portfolio consists of MagMax branded products that use magnetic beads to isolate and purify nucleic acid, that is RNA and DNA, from the sample. This can be carried out manually or automatically. We have a few options for automated sample preparation depending on the throughput of the lab. The user can purify nucleic acid from up to 96 samples at a time using our Kingfisher Flex or Apex instruments. We also provide an internal positive control called Xeno that allows the user to monitor any inhibition in the sample and increase confidence in their data analysis. On the detection side of things, we have our VetMax branded real-time PCR reagents that target the specific sequence of pathogenic DNA or RNA we are looking for. We provide the amplification tools for this as well, from primers and probes for the specific pathogen to the instrumentation to amplify it, such as the QuantStudio 5 and 7500 real-time PCR systems. There are several diagnostic tools available to animal pathogen testing customers. Each of these has a unique set of advantages and disadvantages that can be highlighted in this example here. This slide shows the infection course of the PRRS or porcine reproductive and respiratory syndrome virus. Depending on when the testing occurs, the results might or might not correlate between technologies. Also, it depends on what the specific analyte is that we are looking for. For instance, if you're using PCR, you're looking for DNA or RNA of the pathogen. Depending on assay sensitivity, you could detect it as early as day 0 of the infection, given that there will be a certain level of viral particles present. If you ran an ELISA on that same day, assuming the ELISA was detecting antibodies and not antigens, you would get a negative signal since the pig has not had enough time to mount an immune response and thus create a detectable level of PRRS antibodies. Alternatively, you would get an ELISA positive quite a long time after the virus has cleared from the body, and thus testing a pig after viral clearance would potentially give you a PCR negative. This shows that it's important to understand the status of that animal or herd holistically to make the right decision based on data generated by each of these technologies. I would now like to give you a more in-depth look at the individual steps within our animal health testing workflow, beginning with sample preparation. The key objectives in this step are to release the DNA and RNA from the various cell types that are present in the sample of interest. These could be animal cells, bacteria, viruses, etc., and the goal is to lyse or break open the cell wall or membrane to release the nucleic acid stored within. The next step is to take the nucleic acid through a purification process to ensure you are not co-purifying proteins or other cellular factors such as inhibitors or enzymes. The overall objective is to achieve the best possible amount of target nucleic acid with the least amount of inhibition. The sample prep process consists of 4 basic steps. Lysis is the step where chemicals are used to break open the cell wall or membrane to release nucleic acid along with other cellular factors such as proteins and enzymes. During binding, the magnetic beads bind the newly released nucleic acid and carry them through the rest of the sample prep process. The magnetic properties of the beads allow them to be moved from one step to the next in order to purify the nucleic acid and wash away all unwanted proteins, enzymes, and other inhibitors. The wash steps achieve this goal by allowing loosely bound debris and factors to wash away while maintaining the binding affinity of the beads to the nucleic acid. The final step in this process is called elution, where the now purified nucleic acid is separated from the beads and transferred to a buffered solution for downstream applications such as PCR or sequencing. It can also be stored for extended periods of time for testing later. Our most recent sample prep reagents are shown here. This is called a MagMax Core Kit, and it can be used on a variety of sample matrices. With it, the user can extract and purify both DNA and RNA from the animal sample using either automated or manual protocols. Let's talk a little bit about the components of the MagMax Core Kit and what role they play in sample preparation. The lysis binding solution will lyse cell membranes and viral envelopes so that the nucleic acid contingent in that cell can be released into the solution. It will also denature proteins and enzymes like RNase and DNase so that the extracted nucleic acid is protected. The magnetic bead/proteinase K mix is known as bead mix. The magnetic beads bind the nucleic acid while the proteinase K denatures proteins. Wash solution 1 is a high salt wash that further denatures proteins and washes away inhibitors. Wash solution 2 is a diluted version of wash 1 with a higher concentration of ethanol to wash away the high salt buffer and enhance the binding of nucleic acid to the beads. The elution buffer separates the nucleic acid from the beads and acts as a storage solution for the purified nucleic acid, or RNA and DNA. This slide shows some of the sample matrices that the MagMax Core Kit can process. This list is, of course, ever expanding as customers try new sample matrices such as environmental swabs and air filters to detect low amounts of pathogenic nucleic acid. But this shows the most common sample types that are used during molecular testing. This slide shows an example of the decision matrix the customer would use to choose the appropriate sample preparation protocol depending on the sample and the pathogen type. As you can see, most of the samples can be processed using either the simple or the complex workflow, and the difference is just the upfront processing of the raw sample and doesn't add a significant amount of time to the overall workflow. The idea behind this is to make the process more universalized so the customer only has to purchase 1 or maybe 2 modules from this kit in order to process a large number of sample types. We have a few options for automated sample preparation. One of these is the Kingfisher Flex instrument. It is a magnetic bead-based sample purification instrument that can process 96 samples during each run. This eliminates the need for hands-on sample preparation and allows the user to perform other tasks such as setting up for PCR during the approximately 30-minute runtime. It also reduces the risk of cross-contamination during the procedure and is essential for high-throughput sample processing. This graphic here shows how the Kingfisher actually works. Each of the 96 magnetic rods dips into each well of the 96 well plate and can either mix the sample with the specific reagent, which is when the magnetic rod is separated from the tip comb, or collect the magnetic beads and move them from one reagent to the next, which is when the magnetic rod and the tip comb join. A quick note on an important aspect of the workflow. The Xeno control is our internal positive control and is designed to go through the entire workflow along with our target nucleic acid so that we can monitor if there's any inhibition or other significant effect that could impact our data. Xeno is a synthetic oligo and it comes in either DNA or RNA flavors, and it's designed to be different from any other sequence found on Earth. Therefore, there is very low risk of it being detected by another assay and causing a false positive signal. Since we know the exact amount of Xeno we are spiking into the sample, we know how much to expect after PCR, and so if that concentration is affected by inhibition or some user error during the workflow, that will be indicated in the Xeno data. Xeno has been designed to mimic the target DNA or RNA in terms of effect of inhibitors, so if our Xeno comes up outside of the range we expect, we can assume that our target DNA or RNA has been affected the same way, and can then use that information to troubleshoot or make a call on the data. Now let's get into our targeting system, which is real-time PCR. Here we are trying to target the needle in the haystack of nucleic acid that we created during the sample prep process. There are many different types of PCR or polymerase chain reaction technologies. Real-time PCR is one of them that has been used effectively for years to do pathogen testing. Since the COVID pandemic began, real-time PCR has been in the news and more people are familiar with the terminology now. Basically, real-time PCR allows the user to monitor a PCR amplification in real time. Real-time PCR uses essentially the same components as traditional PCR, such as double-stranded DNA as the target molecule, primers, DNTPs or deoxynucleoside triphosphates that are the building blocks of DNA, PCR buffer, DNA polymerase, etc. The main difference between real-time PCR and traditional PCR, however, is that it uses a fluorescent reagent to the mix so we can monitor amplification in real time. The basic principle of real-time PCR is that the fluorescence will increase as amplification occurs. The instrument we use for this will detect that increased fluorescence after each cycle and record it for analysis. Our specific flavor of real-time PCR is known as TACMan real-time PCR, since we use a DNA polymerase known as Taq polymerase. For this to work, we need at least 2 primers, which are just small pieces of DNA that are complementary to our target sequence. We have one forward and one reverse primer, each sitting on one of the 2 DNA strands. In addition, we have a third oligo known as the probe, which is designed to be complementary to a sequence between the forward and the reverse primer. The probe is labeled with two dye molecules, one, a reporter dye that sits on one end of the probe, and a quencher dye that sits on the opposite end. The quencher dye is a low-energy molecule, and the reporter dye is a high-energy molecule. Now, when these two molecules are in close proximity to each other, in other words, when they're attached to the same probe, when light is shined on the probe, the reporter dye gets excited and releases photons that are transferred to the quencher due to the close proximity. Therefore, there is no net fluorescence released from the system. This is known as FRET, or fluorescent resonance energy transfer. Now, if this probe were to be split in half or cleaved, then when the fluorophore was exposed to light, the quencher would be too far away to absorb the photons emitted by the fluorophore, and thus the photon would create a fluorescent signal that would then be detected by the camera in the machine or the real-time PCR instrument. The basic idea here is that whenever a new product is created from the amplification process, a reporter molecule or fluorophore should separate from the quencher. This way, the fluorescent signal will increase proportionally with the product. Now we will look at what happens at each step in the real-time PCR process. The first step depends on whether you're starting with RNA or DNA. Since PCR requires DNA as the template for amplification, we need to convert RNA to DNA, if that's what we are starting with. If you're starting with DNA, this step is not needed. To convert RNA to DNA, we need an enzyme called reverse transcriptase. At 45 degrees centigrade for 10 minutes, the RT enzyme converts all RNA molecules into DNA. Our next step is to activate the Taq DNA polymerase. This is essentially the Xerox copy machine for our DNA target and is used to make copies of our target of interest if it's present in the PCR soup. The Taq polymerase is heat activated so we can control when it starts doing its job. We have to heat it up to 95 degrees centigrade for 10 minutes to break the chemical lock that we have in place. Now we begin our actual PCR cycling. Each of the following steps will repeat 40 times in the PCR process. Denaturation of the DNA is the step where we separate the double-stranded DNA into individual strands to use as our templates. We can call one the top strand and the other one the bottom strand. At 95 degrees centigrade, DNA will essentially unwind and separate or denature to present both strands separately. Now comes annealing, where we slowly cool down the reaction so that the primers and probes that are in the mix can start to bind to their specific targets on the top and bottom strands. Here we are cooling the reaction from 95 degrees centigrade to 60 degrees centigrade. Now during this annealing process, the Taq polymerase comes in and finds the double-stranded area on the otherwise single strand. This is the reason we have primers, so that the Taq can start its DNA repair from the location of the primer towards the 3' end. Notice that the reverse primer has settled on the bottom strand and the probe is now attached in the area between both primers. Once the reaction hits 60 degrees centigrade, we hold it at that temperature for 1 minute. This is called the extension step, and this is where the Taq polymerase repairs each DNA strand using the dNTPs that are floating around in the soup. It quote-unquote reads the strand as it passes over and signals for a specific complementary dNTP to attach to that point. It's extending towards the probe at this point, and at some point, the Taq polymerase will bump into the probe that has attached to one of the strands. Now, Taq polymerase has a special functionality called exonuclease activity. It can digest any double-stranded DNA that's bound to the template as it is repairing the strand. When the Taq polymerase encounters the probe, it uses that exonuclease activity to chew through the probe and continue to repair the DNA strand. Now look what happens when it starts chewing through the probe. The fluorophore is now released and moves away from the quencher. Now, since light is constantly shining down on this reaction, the fluorophore gets excited and is able to release energy in the form of a photon that cannot be absorbed by the quencher since it's too far away. The Taq goes along and continues to repair the single strand, finally forming an exact copy of the original double-stranded DNA. The same thing is happening at the bottom strand, so that at the end of the cycle, you now have two copies of your template. After each cycle, there is a theoretical doubling of your target in each reaction, and that fluorescence increases proportionally. This slide shows the basic interior design of a real-time PCR instrument. You have your white light source on the top that provides light to excite the probe. You have a variety of filters that absorb and reflect different wavelengths of light. And this allows us to use multiple fluorophores in the same reaction to detect multiple targets at the same time. The plate at the bottom sits on a thermal cycling block that takes each reaction through the different temperatures we discussed in the previous slide. When light from the probe is emitted, it is captured by a CCD camera that converts that light into a signal observable on a graph as a fluorescence number. Once that number is crunched by the computer, it forms a real-time amplification plot that many of you should be familiar with. Here's an example of how the fluorescence is captured by the computer and graphed out. It is very similar to a bacterial growth curve. The geometric phase is where the PCR components like primers, probes, dNTPs, and Taq are in excess. So each cycle creates a doubling of the product. The linear phase is when these PCR components start getting used up. We're starting to run out of stuff to make copies, so the amplification slows. The plateau phase is where all the PCR components are finally exhausted and amplification stops. So depending on how much starting template you have, you can imagine that this graph would start earlier or later. This last section deals with data analysis and how we make sense of real-time data. This is what a real-time amplification plot looks like. It may not be as neat and clean as this, but essentially, this is what the user will be presented with after the run is complete. The graph is plotted with fluorescent units or delta Rn on the y-axis and cycle number, a function of time, on the x-axis. Some terms that are used during data analysis are shown in the next few slides. Baseline is the initial signal level where amplification is not really affecting the fluorescence level significantly yet. This setting essentially cancels out any noise during the specified cycle range, and if set incorrectly, it can cause issues with the background noise being interpreted as actual data. Threshold setting is of significant importance because it determines where noise becomes signal. Any amplification signal that crosses the threshold will generate a CT or cycle threshold value that determines how concentrated your target is in the sample. If the threshold is set incorrectly, it could lead to false positives or worse, false negative data that could be very detrimental to downstream decision making based on this data. There are a few different ways to set the threshold, and I will describe the way we usually recommend setting it. This is called control-based thresholding. The control-based threshold, or CBT method, allows the user to set the threshold based on a predetermined percentage of the maximum fluorescence of the positive control for that specific target. It is definitely not an automated setting like the auto CT function, but for our animal health applications, it is advantageous because it increases consistency between runs, technicians, and labs, and it can remove any variability around lower fluorescence from one machine or plate to the next. It also reduces the occurrence of aberrant curves that can occur if using the auto CT setting. Finally, we will talk about a few challenges associated with real-time PCR data analysis. There are a few reasons why a late or weak signal could occur, and some of these causes are shown here. Is it contamination from another well? Is it a software anomaly? Is there inhibition in the sample? Worst case, is there a mutation in the target region that's causing low amplification efficiency? Or is it the obvious low viral titer? The good news is that we have controls in place to verify most, if not all, of these observations. We have negative controls for contamination check, multi-component graphs to determine true versus false amplification, Xeno for monitoring sample inhibition, and for mutations in the target, we can certainly see a trend from previous data or rely on other tests and clinical signs to determine if we are dealing with a mutated strain. For low viral titer, running replicates or retesting is the only good way to determine a positive from an accidental splash or another one of the conditions in the previous slide. The more sample you input at the beginning, the greater the chances of capturing low concentrations of the target in your PCR. This graphic shows an example of different concentrations of target and how they would impact the amplification curve. High copy numbers of target mean that whatever volume you pipette, you should have a good chance of getting a detectable level of your target. Medium concentration should be detectable as long as you have enough of your sample represented. And as you can see here, low concentrations of your sample would require a lot of sample or multiple replicates to catch the template. We certainly have tips and strategies in place for any customer interested in getting the most out of their data, and there is certainly enough information to share that it would require its own presentation. Thank you so much for your attention during this hopefully informative presentation of our animal health pathogen testing workflow.
Hello, everyone. Thanks for joining us in the live Q&A session. I want to thank Rohan again for being here in person so he can get all your answers. And then while we're waiting and accumulating some of your questions, we're going to be collecting a poll as well. So feel free to fill that out real quick, and we'll be back in a second with your questions. Feel free to enter them in the chat. All right, I think we've had a couple of questions come through, so I think we'll get started here. So thanks for joining, Rohan. I'm gonna dive in real quick and see, what kind of questions we can get answered while we got you on the line.
Sounds good.
Regarding the sample extraction, what is the typical extraction to amplification timing in this workflow?
Yeah, so it certainly has gotten shorter. First of all, can you guys hear me? Can
Yep.
everybody hear me? Okay, cool. That's a good question. So it's certainly gotten shorter over the years as we've optimized our workflows and made our reactions and reaction chemistries faster. But I would say typically around 3 to 4 hours is generally how long it takes from the time you get your sample to the time you put it on the Kingfisher or however you manually extract it, and then by the end of PCR, it's about 3 to 4 hours. But again, that's kind of variable. It sort of depends on the type of sample. It depends on the number of samples, and it certainly depends on your extraction method. Automated being a little bit more hands off for the user versus manual being more hands on. So depending on kind of time management there. But I would say on average, it's about 3 to 3.5 hours.
Awesome. No, that's really helpful. With you kind of mentioned the instruments there in Kingfisher, but there's a question that came through that which instruments are most commonly used with your reagents? I was wondering if you could speak to that real quick.
Sure. So, we have two main sections of our workflow. It's the sample preparation part and then the detection part, which is our real-time PCR. And our chemistry is based on magnetic beads. So we have instruments that are kind of specific to that chemistry. There are multiple different kinds of sample prep chemistry, but we have magnetic beads. So we tend to use the Kingfisher line of instruments, which are essentially just large automated magnets that kind of move these magnetic beads from one step to the next. So depending on your throughput, you would be using the Kingfisher Apex, our latest one, or the Kingfisher Flex for your larger throughput, so 96 samples at one time. The Kingfisher Duo Prime would be lower throughput, so you'd be able to process 12 to 24 samples at a time. And then on the PCR side of things, again, we have a couple of different options. Our sort of workhorse for the last many years has been the 7500 fast, Applied Biosystems 7500 fast instrument. But we have a newer generation, the QuantStudio line of instruments, and the QuantStudio 5 is sort of the direct replacement to the 7500. And those instruments also can process 96 samples at one time.
Yeah, I think a lot of us are very familiar with the 7500 fast being, the tried and true workhorses of the AB program. But it's nice to see, some new innovations coming through, especially with the QuantStudios and then also the Kingfisher Apexes as well. Really cool stuff going on there. With regards to training, how easy is it to be trained on running, a PCR test or the workflow as a whole?
Yeah, that's a good question. It certainly depends on your initial kind of skill level before you come into the training. So if you're familiar with lab equipment, your general lab equipment like pipettes, reagents, test tubes, using freezers, that kind of stuff, then I mean the training itself is really mainly just being able to pipette small quantities of liquid from one tube to the next. I'm sure it's hard to kind of visualize what's going on in a lot of these reactions because it's all happening at a molecular level. But certainly the theory training is sort of what takes a little bit more understanding, and work. But as an industry, we've created these kits that are just super easy to follow, kind of a workflow as long as you have a few essential skills like pipetting. So as long as you can follow a protocol, it's fairly easy to get through some of these kits, and we are always striving to make them easier for the user to use and also more universal so that they can be used for a variety of sample types and other applications. So like I said, it's easy if you have a predefined skill set for sure. And I mean, personally, I've trained people with all different backgrounds, and I think people are able to pick up on it fairly quickly. And like I said, it's getting simpler and simpler as we develop more and more universal and easy to use workflows.
Yeah, no, I was going to say, in regards to training, I feel like you're the person to speak to over anyone. Probably done this, 100 to 1,000 times, but it's good to hear that the complexity, isn't too high of a ceiling. Most
Right.
of us, I would hope, can work a pipetter here and there, but in terms of the investment in training, you know, compared to culture or other technologies, how does PCR consume department resources, financial or manpower wise? How many people does it take to really run these tests?
Right. So PCR, I mean, it's kind of been the gold standard for pathogen detection for a decade or more at least, but certainly has been an expensive resource for a lot of customers and companies to come online with. Obviously, there's a huge number of advantages with that too, in terms of sensitivity and a lot more automation and the amount of information that you can get from just one reaction with multiplexing and stuff like that. So it's kind of a balance there, but certainly compared to culture, at least, I mean, culture is kind of the baseline, for detecting a pathogen. It totally depends on what you're detecting. So say, for instance, you're trying to, grow MAP or, a mycobacterium, something like that, which is a very challenging, pathogen to grow up. It could take days or potentially even weeks under the right conditions. And if they aren't the right conditions, then you could potentially have, a false negative, for instance. So, if culture is super cheap to work with, I mean, those plates and the media and all that kind of stuff is fairly inexpensive, and the skill level required to just learn how to culture a plate is fairly reasonable. So a lot of people can do culture, but again, then you're kind of giving up on sensitivity and potentially spending a lot of time trying to grow up a plate that may or may not end up being successful in its growth. So that kind of just depends. There are certain situations, of course, where culture is still the gold standard, but more and more people are now getting familiar with PCR and PCR techniques, and they are getting cheaper too as more and more the industry adopts it more and more. So it's certainly definitely a balance that needs to be met between finance and your advantages of PCR over other technologies.
Yeah, and I think you said it well there, where everything has its place in terms of applications. But with a lot of things that our customers are working on, and people within these laboratories, I think the turnaround time is very important on a lot of these tests. So I think PCR really gives an advantage to that field to make sure that our customers are providing the fastest and most timely results to help get herd protection at a good level. So that's good.
Exactly. The sooner you can detect your pathogen of interest, the sooner you can take action. So that's certainly a very big consideration.
Could not have said it better myself. So I'm not seeing any other questions left in the chat. So Rohan, I'm gonna leave it up to you. Is there anything you'd like to say to the viewers before we call it quits here?
Just thanks for your attention. Thanks for listening. And I hope everybody's learned a little something. I always feel every time I give this talk, whether it's pre-recorded or not, I always find that I'm learning a little bit more about this process. And hopefully, if there's any other questions that come up, feel free to reach out to one of us, and we'll try and get those answered as soon as possible. So thank you very much.
Could not have said it better myself. Thanks so much, Rohan. I really appreciate you hopping on here. I'm sure many people as well feel the same sentiment.
I think we lost you, Charlie.
Oh, well, on that note, I think we'll we'll end the recording. But thanks so much everyone for joining, and thanks again to Rohan for giving the presentation.
Thank you.